Hey guys, my name is Molly and today I'm going to be presenting on diphtheria. Just as a warning, diphtheria is a bacterial infection, so there are going to be some pretty gnarly images coming up. Okay, so to start off, what is diphtheria? It's a bacterial infection, like I said. It's toxigenic, meaning that its mode of infection is that it destroys healthy tissue by secreting a toxin, and there were really high morbidity rates of diphtheria pre-vaccine. Its hallmark symptoms are a thick, gray, membranous coating over the back of the throat and tonsils, or you can also get it on your skin where a similar sort of ulcer will appear. Some history and background of diphtheria. It was supposedly first described by Hippocrates in the 5th century BC, but since it was so difficult to distinguish from scarlet fever, we see really prevalent recordings of diphtheria happening around the 1600s. So that's when it's first described. It's named in 1826, it's identified in 1883, and by 1890 we have the antitoxin discovered, and an antitoxin just neutralizes the effects of the toxin. I don't have time to go into it now, but it's a really interesting story if you want to look into it. The first vaccine comes out in the year 1920. Diphtheria rates start to drop, but not as rapidly as until the 1940s, when the vaccine for diphtheria is combined with that of tetanus and pertussis and becomes widespread in the United States. So diphtheria rates go way down. Between 2004 and 2009, no cases in the United States. Isolated outbreaks can happen. One happened in 2015 in Spain, which is also an interesting story that you should look into. But in general, in most Western countries, diphtheria is no longer a huge issue, but globally it does still occur, and in 2016 there were about 7,000 cases. For some incidence and prevalence, pre-treatment, so pre-1920s, there was a 50% morbidity rate. At this time, it was the third leading cause of death in children in England and Wales. But since the vaccines have been being produced now, it's really only endemic to subtropical regions. Some statistics for you guys. In 1921, there were 206,000 cases with about a 7.5% morbidity rate for those. That morbidity rate is still the same today. So between 5 and 10% of all people who contract diphtheria, even with treatment, will die. And that number jumps to about 20% in individuals younger than 5 or older than about 50. But in the United States, since 2004, there have only been about 2 recorded cases. So now that we know some history about it, let's talk about the technical stuff. What is the bacteria that causes diphtheria? So its classification is, its infectious disease name is diphtheria, as I mentioned, and the causative agent is Corynebacterium diphtheriae, and here's a picture of it here on the right. Some characteristics of it: it is a bacteria, not a virus or protozoa. It is bacillus, non-motile, non-capsulated, gram-positive, and aerobic. Now that we have that out of the way, we can get to the interesting stuff: how it's transmitted and what it looks like. Transmission happens most readily through the air, so through coughing or sneezing, those airborne droplets get suspended. It's also transmitted through deposits of a person's saliva, so on a drinking glass or a towel or a tissue that they might have touched. Since you can also get diphtheria not only in the back of your throat but on your skin, you can contract it by touching someone's infected wounds, and infected individuals can spread diphtheria for up to 6 weeks. These different modes of transmission lead to outbreaks. So there are 2 types: large scale and isolated. First of all, it's, like I said, it's only endemic in subtropical regions still. Large scale outbreaks will occur in unvaccinated regions where there are crowded conditions and poor hygiene standards. But isolated outbreaks can still happen in countries with strong vaccination programs. What will usually happen is that someone will travel to a subtropical region, bring it back with them, and if they live in a crowded or poor hygienic area, then an isolated outbreak will occur. So now that we know where it's happening and how it's transmitted, we can get to our two types, which are respiratory and cutaneous. So like I said, back of your throat, nasopharyngeal cavity, or on your skin. The symptoms of the respiratory diphtheria begin 2 to 5 days post-infection. You got a lot of flu-like symptoms, fever, chills, malaise, nasal discharge, swollen glands. But these swollen glands are really important because in diphtheria, they are extreme. They become so swollen that it results in a condition called bull neck, which you see here on the right. And finally, there is the formation of a thick gray membrane covering the throat and tonsils, which can lead to some hypoxia, difficulty breathing, and pain swallowing. You can also get it on your skin with some of that same pain, redness, and swelling associated with bacterial skin infections. And what it is really just an ulcer covered by a gray membrane. Okay. Now we can move on to our next section, the behind-the-scenes stuff. How exactly is this bacteria getting into humans and infecting? So the pathology, we have our affected tissues are the tissues of the nasopharyngeal region or throat or the skin, and there are lesions that develop on these areas. If left untreated, these symptoms can escalate into breathing problems, heart problems, or nerve damage. You can recover even from these complications with treatment if diphtheria progresses to these advanced stages. Not all hope is lost. It's okay. Okay. So for our pathogenesis, how exactly is it damaging the cells? So the Corynebacterium diphtheriae cells will colonize either the throat or the skin and form that lesion that I mentioned, and then they'll start secreting toxin. The toxin destroys healthy tissue and leads to the buildup of this gray pseudomembrane, which is made up of dead tissue, bacterial cells, inflammatory cells, and some fibrin. However, not all strains will do this. So let's take a quick sidebar onto strains. There are two types of strains of diphtheria, toxigenic and non-toxigenic. They can all produce the toxin, but the non-toxigenic ones just tend not to. They can convert to toxigenic strains and they can start producing toxin, which is why in the lab it's important to test all strains. And then diphtheria is also the paradigm of toxigenic diseases. It's really the model of study when looking at infectious diseases that infect via toxin. Okay, so back to our pathogenesis. There are two main factors of virulence that it uses, colonization and toxin production. So it needs to colonize the area, and then it needs to start producing the toxin. The toxin is extremely potent. A little bit goes a long way, and that is what is more heavily studied. Not really much is known about the colonization. The toxin production, what will happen is that there are 4 distinct steps, which I don't have time to get into now, but what it boils down to is a type of endocytosis. And once the toxin gets into the body's cells, it inhibits protein synthesis. That's how it damages. Okay, now onto the response stuff. So what happens once an infection is present? So first we'll start with immune response. It's a humoral response, so you'll get an antibody response following the disease or the immunization. The primary protection is from immunoglobulin G class antibodies, and also there is phagocytic activity associated with recovery at the infection sites. Since it's a humoral response, little is known about the cell-mediated immune response. On to diagnosis. So the main thing that physicians will look for is that gray pseudomembrane, but once it is found, diphtheria does require lab confirmation that the bacteria is present. And clinicians also have to be careful that they are swabbing the actual lesion and not just the pseudomembrane, so sometimes that has to be cut to get underneath it. And you can see here on the right, there are a variety of different agar tests to undergo. To prevent it, sorry, it says prevention, just my screen is covering it. To prevent diphtheria, The best thing to do is a vaccine. It's highly effective, the stats show that, and also hygiene and crowd control, as we've talked about before. To treat it, it is treated with both antibiotics and antitoxin. And I have some pictures of horses here because the antitoxin is made from horses, and I wish I could talk about that, but I don't have time, so look it up. And finally, to infinity and beyond, the research and future outlook, what the future holds for diphtheria and research about it. So on the research side of things, the research status of diphtheria is that they are making GMO toxins from it. So there are 3 distinct regions of the toxin peptide chain. There's the catalytic section, the transmembrane, and the receptor binding section. Scientists are currently studying the receptor binding region. They are editing it out, and they are putting in different domains from hormones or cytokines, and this makes a chimeric protein, and these are called fusion toxins. The great thing about fusion toxins is that they will only intoxicate the cells with the correct targeted receptor. So what that looks like in the future directions for diphtheria research is that these fusion toxins are being used to treat autoimmune diseases and refractory lymphoma. The first, I like to call it genetically modified fusion toxin, is being clinically tested on humans right now to help treat these, like I said, autoimmune diseases and refractory lymphoma. I didn't actually know what refractory lymphoma was. It is a type of lymphoma that has not responded to the initial treatment. So this is pretty exciting because it looks like diphtheria toxin-based fusion toxins, kind of a mouthful, I know, sorry. These fusion toxins will become extremely important in treating disorders in which specific cell surface receptors may be targeted, or in short, these fusion toxins will become the tumor treatments of the future. Here are my references, going to be expounded in my paper, and I really hope you enjoyed my presentation and learned a bit about diphtheria. Thanks.